I want to talk a little bit more about Microsetta, where we're going with Microsetta, and what kind of some of the bigger pictures are with the project. But first, we need to step back a little bit. And also, I'm required by the University of California to tell everybody that I have a conflict. I own equity in BiomeSense. But 10, 15 years ago, there was this big project called the Human Microbiome Project, and we collected samples from about a couple hundred individuals, and we were able to derive this map of the relationships of microbial communities among various different body sites. As one of the things that stood out from this project, though, was that nobody could just participate. You had to be a very special individual to take part in the Human Microbiome Project as a whole. So we launched this effort called the American Gut Project, where we allowed anybody to contribute some money, and in exchange, we would send them a sampling kit, and they could give us some poop. And from that poop, we would generate a report that they would get back and give them a little bit of an idea about what was going on within their microbiome. And this led to a extensive research database, which has gotten use in projects like Borderlands Science, as well as many other projects behind the scenes to kind of provide a backdrop for understanding a little bit more about microbes and how they relate to us and our health. But it's also been well known for quite a long time that geographic variation is a very strong factor in differentiating microbial communities that could be far greater than the variation that we observe among different important diseases. To visualize this a little bit further, I'm going to take a brief jaunt into a meta-analysis where we've combined 16 SV4 data from a variety of different studies from many different countries. And we're going to start off first by looking at the Yatseneko et al. Dataset, the global gut dataset, which was from 2012, where there were fecal samples collected from Malawi in the United States and Venezuela. And there was this huge geographic separation that was observed within this data. Now, if we add in these other countries, you might not be surprised to learn that they fall all over this map, where we tend to observe Western populations grouping together, non-Western populations grouping separately. One of the fundamental issues, though, and I think challenges for the microbiome field, is how do results collected in one population translate into what we might observe in another population? So this has led us to a big shift in what was the American Gut Project, which is that, well, most of the world is not America. So we wanted to create a more flexible framework for finding opportunities for people to participate. And we launched an effort called the Microsetta Initiative, which is led by Dr. Sejin Song at the Center for Microbiome Innovation. So why the Microsetta Initiative? Well, we all have these modern Rosetta devices that sit in our pockets nowadays that do these fancy things like translating meals in one language or menus in one language into another. And we thought it would be really cool to try to figure out how can we translate poop from different countries into consistent results so that we might be in a better position to for example, provide actionable information about like how you might be able to change your diet, but also what does it mean to be, for example, have IBD in one population, and is there consistent signatures within another population? So to that end, the Microsetta Initiative has a discovery workflow where we can work with other groups to identify and create cohorts. We can have targeted cohort engagement. And this actually goes back to touching on the work that Patrick Vega opened up on that was done in collaboration with the No Nutritia research to reach out to many populations around the world, perform qualified analysis, and generate an analysis reporting system. Our solutions in this space include validated collection devices using 95% ethanol, which is molecular grade and also suitable for untargeted mass spectrometry and shelf stable for upwards of a month or two, so suitable for room temperature shipping. And there's a bunch of molecular work behind the scenes that also standardizes how we do the processing in lab to generate high quality results. We also allow tailored surveys, participant generated responses, and customized surveys for individual specific sub-studies that might utilize this platform. The cohort creation can really take two different arcs. we can dual consent in an IRB approved manner, dual consent with other projects to establish new groups to participate using the Microsetta framework, or we can also go back and screen our existing population base, which at this point is over 30,000 subjects, and potentially identify interesting groups of people to recontact. Through this, we can engage with people with surveys about what we have observed in the microbiome samples. And so this is a little bit of a different arc from, from some of the projects that Patrick was talking about, where we do have ethical approval to provide some degree of information back to participants in exchange for their contributions to the project, although there is no medical information and no medical advice provided along the way. And all of our data analytics are performed through automated open source pipelines, which they themselves, many components of, have been independently published and peer reviewed in high tier journals. And then while working with the group, we can, of course, gather all of the data that was collected for a given sub-study and directly integrate it with the broader data set, so we can minimize any types of technical concerns in order to leverage the significant background that we have generated throughout the course of this project. Our kits at this point are optimized for fecal and skin samples. There is a lot of metadata that is produced from these subjects, in addition to validated food frequency questionnaires. Our protocols are published, benchmarked. There is a huge engagement, and we also have capacity now as well too for longitudinal collections, not just for somebody submitting multiple samples, but for collecting updated participant survey information or dietary information along the way. One thing that's not noted here is these data are almost entirely open access for anybody to use and reuse, as we saw, for example, within Borderlands Science, where some of the original results from American Gut were made available for a what I would characterize as a very fun and interesting gamification project. So to give you an idea about the technical consistency of this project, so we've been running American Gut and Microsetta for over a decade. We've done over 150 different sequencing runs. And what you're looking at is each successive sequencing run being added into a common data frame. If you squint in the upper left, you're going to notice that we're going to go from 16S to shotgun data and in back and forth. And so this is made possible through some novel developments we've made through the Green Genes 2 phylogeny, which bridges whole genomic and 16S data and allows us to unify technically disparate sequencing protocols, which is, I think, a quite wonderful development in this space, because it allows us to harness the existing data that not just exist in an effort like Microsetta, but in other studies throughout the world and other important areas of focus. So if you want to learn more, you can go to our website, microsetta.ucsd.edu. It provides some information on contribution. And as excited as I am about reaching out to the rest of the world, we are still in a growing phase with our new collection kits focused on the US, but we have a keen eye for strategic expansion, and it is fundamental to the project that we find creative ways to engage with populations outside of the US. And that is a constant area that we are working on. There have been many people involved in this project, from graduate students and undergraduates and technicians up to postdocs and faculty members. And many people have used this project in a variety of different ways to help advance their own careers. The project has, for the most part, been funded off of individual participant contributions. However, there has been some phenomenal work supported by Danone Nutricio Research, in particular, actually, when Patrick Vega was at Danone, and he helped get some of those projects off the ground. And if you would like to learn how to get involved, you can, of course, go to the Microsetta website. The data are all on our platform called Cheetah at UCSD, and you can always reach out to me if you've got any questions or curiosity, or would be interested in trying to identify a collaborative opportunity to harness the Microsetta infrastructure for your purpose.